Welcome back to video lesson 5, part 2. This part's optional unless you were away. Otherwise, we will be covering this material in class. So now we're going to look at the various structures of carbohydrates. In particular, there's 9 structures that you need to know. If you're someone who enjoys the game in the newspaper where they show the 2 pictures and you have to spot the difference, this will be fun for you. If you don't enjoy that game so much, unfortunately, you still need to know these structures. So we're going to begin with 3 key monosaccharides. And just to remind us from before, mono refers to one or single ringed sugars. In this case, we're going to be looking at 2 different shapes of rings, those called pentose, because they have 5 sides, and those called hexose, because they have, you guessed it, 6 sides. And just to highlight here, when the name of something ends in ose, it generally indicates you're working with a sugar. So the first one we're going to look at is called fructose. You've probably heard of fructose syrup as it's the #1 sweetener used in food today. Unfortunately, our body doesn't respond to it as well as glucose, but it's cheaper to make, so many of our foods have it. So fructose, if you look carefully, 5-sided ring. So it is a pentose sugar. And it's found in fruit. Now you guys do know already of some other 5-sided sugars from your DNA back in grade 11, ribose and deoxyribose. But the difference here, if you look carefully, is in ribose and deoxyribose, you only have this top CH2OH group, whereas in fructose, you also have this bottom CH2OH group. And in fact, even though it's a 5-sided ring, a pentose, it is a pentose that has 6 carbons. You can go around and count each vertice is a carbon as well. So there's 1, 2, 3, 4, 5, 6. The next sugar we're going to look at is glucose. I think you can see one obvious difference right off the bat from fructose, but we also have to compare glucose to galactose, so I'm going to give you a second to see if you can spot the difference. You spotted it? Hopefully you have. The key difference, even though they're both hexose sugars, is that if you look at glucose, the left and right OH groups, or the edge OH groups I like to call them, are down compared to this top CH2OH group. So the way you recognize it, edge OH groups are down relative to the CH2OH, okay, or on the opposite side as the CH2OH. Now, glucose is extremely important. Some people argue it is the most important monomer, at least sugar monomer, most important sugar monomer. In human body. As it is used to make ATP. Which we mentioned briefly before is the energy currency of the body. you want to get anything done in your body, you need to spend ATP. In contrast, if we look at galactose, we look at its edge OH groups, you'll notice they're up or on the same side relative to the CH2OH group. So, edge OHs are up. Now this may seem like a subtle difference. What does it really matter? Well, the way our body reacts to it is very different than the way our body reacts to glucose. Very minor differences in structure can have huge impact on function within the human body. We're going to see that theme continued throughout Biology 12. So in this case, what is galactose? Well, it might remind you of a sugar you're more familiar with, lactose. It is actually a monomer. Remember, that means a building block of lactose, which is a more complex sugar found in milk. So, galactose is found in milk. Next, we're going to look at 3 key disaccharides. And although there's lots of different disaccharides in our body, these 3 that we're going to look at have in common that they all contain a glucose. So let's take a look. Remember that di refers to 2 ringed, the saccharide, the ringed sugars. In this case, all are made, not all disaccharides, but all 3 that we're looking at are made from glucose plus one other monosaccharide. And how do they come together? They use a reaction we learned about last day via a dehydration synthesis. So if we look at the first disaccharide here, look carefully, see if you can spot the glucose. Pretty simple in this case because there's only one 6-sided ring. Here's our glucose. Plus, you should recognize the other ring, or start to recognize it. The pentose sugar that we've talked about that has a CH2 group both up here and down here was fructose. And when you join those 2 together, we get a sugar that many of us are fond of, and it's why we enjoy eating fruit, sucrose. So this is found in fruit. Gives it its nice sweet taste. The next disaccharide we're going to talk about, and the one you really are going to get very familiar with and need to know quite well. Okay, let's spot the glucose. Here's a glucose. And if you look really carefully, over here is actually another glucose. So it's glucose plus glucose equals a disaccharide called maltose. And maltose is generally the first digestion product of polysaccharides. So when we consume a polysaccharide, our body will break off dimers, two of these, of maltose, break off maltose, and then maltose will be further digested down into glucose. The maltose product will then be further digested down to glucose, which our body can then use effectively. And our third disaccharide we need to know about, down here, if you look carefully, okay, OH group is up here. That would make that galactose, plus OH groups down here, that would make that glucose. When you get glucose plus galactose, that is lactose. And some of you may be lactose intolerant. And what this means is that you lack the enzyme necessary to break down the bond between the galactose and the glucose. So you can't digest lactose. And then it builds up in your system and causes all sorts of unpleasant side effects. Now here's the interesting thing. If you drink milk regularly, most people will continue to produce the enzyme necessary to do this digestion. But if you stop drinking milk, then you stop making the enzyme, and then if you try and go back to drinking milk, you're more likely to feel like you're lactose intolerant. Of course, you can take something called lactate, which helps you break down lactose so you can continue to consume milk even if you're lactose intolerant. And we're just going to go back up to the top here where we have a little space, and I'm just going to quickly show you how this bond forms via dehydration synthesis. What happens is you have your OH group here, and then your glucose molecule. You have your OH group here, and you have your fructose molecule. It's a pretty bad pentose, but. And then in the reaction, you get one of the OH groups and the other H coming together to form H2O. And in that process, this O, which is still linked to the carbon here, comes over here and links up here. So you sort of cut out the OH and the H, the water, and link the O between the two monosaccharides. And that equals the dehydration synthesis. And why you end up with this little O right in the middle. Okay, I'm just going to stop it there.